Andy Smith, who will be talking to us about a really interesting topic in MS, Epstein-Barr Virus. So I'll turn it over to him.
All right. So thank you for that wonderful introduction, and we're going to get right into it. So before we can understand what Epstein-Barr virus is and understand how it affects MS and what we can do about it, ultimately we first have to understand what is a virus, and then we'll build from there. So going into what a virus is a virus is a sub-microscopic infectious species. So I put this kind of scale in here to give people an idea of how tiny we're talking about. So imagine you take one of your hairs and you kind of look at it along its thinnest margin. That's about 50 to 100 microns in diameter. So a micron is 1,000 nanometers. So everyone is kind of familiar with red blood cells. Those are the cells that carry your oxygen around your body. Essentially, they're about 8 microns. So they're about 1/6 the size of essentially a hair follicle. Now, even smaller than that, about 25% smaller than that, is a bacteria. E. coli, most people are familiar with that if you don't cook your hamburgers well enough. But there's a lot of other things that cause that. And then we get into some substructure parts of the cells called lysosomes that help the cell function, that are on the order of essentially 1 micron or smaller. And then we get to the viruses. Some of them are about 10 to 1,000, almost 1,000 times smaller, or 100 times smaller than your red blood cells. So they're very, very teeny. So some of you who might be wondering what this thing is in the top upper corner, that's actually a image of the Epstein-Barr virus, which we'll talk about a little bit more as we go along. So because I'm nerdy, I always kind of like to have this debate of what is life. And I'd say as a field, it's debatable whether or not viruses count. So typically, life we consider something that's able to essentially reproduce itself on its own. So the bacteria, classically, everyone would say, is the lowest level of life. But some people would argue that viruses are alive. They have the same genes that essentially, or DNA or RNA that essentially we have, that kind of helps program our body to do things. They can change over time, as we've seen with all the very variants of the COVID virus change over the past 2 years. We can see that they clearly evolve, and they can copy themselves. They just need essentially a host, whether it's our cells, bacteria, or some other type of cell to reproduce itself. However, the reason why a lot of people say that they're not alive is because they are unable to do any of this on their own. So this is kind of the simplest kind of concept of what a virus is. So a virus has to have a capsule or capsid. Essentially, that's the coating around the DNA. Without that, the DNA would just be kind of floating off inert and really couldn't keep itself together. And then the DNA or RNA or the genetic material itself essentially is critical, because without that, the virus would never be able to build any of the things to replicate itself. Now, optional parts are the envelope, which kind of coats. So we kind of see these little protein spikes coming off the envelope. You can kind of see them matching the picture of the real Epstein-Barr virus in the corner. Those are a lot of times something that we'll use to target vaccines towards. And then there's the tegmentin, which is the space in between essentially the capsule and the envelope. So how do viruses reproduce? As I said, they can't do it on their own. So I'm going to kind of go through a 5-step process that's similar to the process that we'll see for Epstein-Barr in a little bit. Step 1 is that the virus has to attach to a cell wall. Then it inserts its DNA into the cell that it's going to infect. And then after it's there, it either has to figure out a way for its RNA or DNA to be essentially replicated and transcribed to build proteins and more RNA so it can ultimately have the components to build a new virus. Then finally, the virus will start assembling itself. And then eventually, when it reaches a critical point, it will start releasing itself from the cell, either through a controlled mechanism or the cell will just rupture because it just is that full of virus. So as I said, there's two main types of viruses, DNA and RNA. DNA is the type of genetic material that we have that makes up our body, that's part of our chromosomes. RNA is typically transcribed from DNA to make proteins, and viruses can be made of either one of these. If it's a DNA virus, it typically replicates in the cell nucleus, and that's where human DNA would be. If it's an RNA virus, it typically uses some cellular components outside the nucleus because it doesn't have to go into the nucleus. So one of the interesting facts is when you consider human DNA, essentially about 8% of it may be viral in origin. So that's how much viruses have been able to affect humans over time by essentially affecting our cells. It stays with us, and then we pass it on even to our family. So going into Epstein-Barr, essentially it is one of the many different types of human herpes virus, and it's on the size of 0.1 microns. So to put that in perspective, that would be 500 times smaller than the width of your hair. So it's very, very tiny, and only really fancy microscopes can actually detect it. It's a DNA virus, so it uses the cell nucleus in order to replicate itself. And it's the most common cause of mononucleosis. So essentially, how does Epstein-Barr virus spread? It more classically spreads through saliva, so sharing a drink, sharing food, sharing, pretty much anything with a person kissing. Mononucleosis used to be known as the kissing disease. And essentially, it is endemic to the population of the world. More than 90% of people in the world have been exposed to Epstein-Barr virus. When you get younger, it's kind of like chickenpox in some ways, where it's typically a less severe form, but most of us have known teenagers, or many of you in this room may have actually had infectious mononucleosis, where you can get severe swollen lymph nodes, fevers, kind of malaise, and that malaise can last for months. And sometimes the lymph nodes and other lymphatic tissue can get so swollen that sometimes people can even get a splenic rupture. So it is definitely a large source of disease burden in the world, and this virus is responsible for many cancers. So it's not as common in the United States, but throughout the world, Epstein-Barr virus is associated with several malignancies and cancers. So how does this work in humans specifically? Well, step one, it has to bind to essentially B cells, and it can also bind to epithelial cells. And once it does that, it enters the cell and causes the cell to enter a latency phase. And sometimes it can go through the cycle very quickly. Other times it can take a while to go through the cycle. And essentially this latency phase transitions the B cell from an active B cell to a memory B cell. So it's a long-lived cell that stays in your body. And essentially it will kind of stay there. And when the cell gets infected with a different virus, that will typically activate the stage where it starts replicating its genome more, starts replicating its virus capsule more, and that's when it will start shedding the virus again. So when it sheds the virus, not only is it actually infectious to anyone who you happen to share a drink with or a kiss, for example, but also it can also infect other B cells in your body. So essentially, it can cause this almost continuous infection that never quite clears. And that's kind of why we think Epstein-Barr stays with a person for as long as it does. So we can do tests to determine how acute your infection is by looking at certain antibodies. So the viral capsule antigen essentially is the antibody that looks at the capsule that covers the DNA. IgM typically means it's a little bit more of an acute infection. IgG means that typically it's been there for a little bit longer in the process. And the Epstein-Barr nuclear antigen kind of establishes more of a chronic phase of the infection. So by doing simple blood work, we can detect, where you would be at in this stage. And there are people that essentially come out with these wonderful graphs or tables that would allow pretty much any doctor to go like, okay, so you're negative for all three. Well, that means that you have never been exposed to this virus. And depending on what one of these factors are positive, we can determine if you have a chronic infection or a brand new infection. We can determine if it's in a latency phase or if the virus has been reactivated. And as I said, that it's found in several cancers throughout the world. More commonly in the United States, it's been associated with lymphomas, particularly Hodgkin's and non-Hodgkin's lymphoma. In other parts of the world, more of the epithelial infection can cause a nasopharyngeal carcinoma, and it's also been associated with gastric adenocarcinoma. So you can actually look at the virus and these markers in tumor cells and kind of figure out what specific tumor it is, and it's a way to kind of determine what cancer someone has. So I'm going to shift gears a little bit and start talking about Epstein-Barr virus and MS. So I'm going to go back in the Wayback Machine of history, and one of the first things that kind of led people to think that Epstein-Barr virus or some infection may have been related to MS is this case, like incidence report of MS in this tiny island called, I'm going to butcher its name, Faroe Island. It's a semi-independent unit of Denmark, and it's in between essentially Iceland and Norway, kind of north of the United Kingdom. And as I'll see in the next slides, essentially prior to World War II, essentially this virus or MS was practically unheard of. So essentially, British soldiers were stationed there in the early or late 1930s to the early 40s. And essentially, there were never these British troops there. It was kind of a very tiny population. The population didn't really have a lot of outside influence. They kind of lived amongst themselves. So essentially, the island had a population of 48,000 people prior. And essentially, prior to the British soldiers being there, the rate of MS was like 0.2 in 100,000. So it just was not really seen there. And then the rate increases profoundly to 5.9 for the first roughly 10 to 20 years after the war. It goes back down, and then later a second generation or second rate of kind of this occurs. And that's one of the things that led people to like, maybe there's an infectious cause to multiple sclerosis. That's kind of one of the things that led into the theories of interferons, one of the earliest class of medications being used to treat MS. And ultimately, more studies came out that further supported this. So this is a relatively tiny study of essentially 2,767 people in southern England. And essentially, they covered all these people who had mononucleosis out of a possibility of like roughly 700,000 people. They were watched for about a 36-year period. And generally, they didn't really see the increased incidence of MS until about 10 years after. Now, admittedly, this is a relatively small incidence rate of multiple sclerosis, being 6 patients out of this large population. But we did see after the infection, there was an increased rate. And this will kind of build on the study that really kind of convinced me that there's probably a relationship between Epstein-Barr and multiple sclerosis. Essentially, this was a German study that retrospectively analyzed MS patients and whether or not they were positive for various serology. So essentially, out of 901 patients that this study did, 380 had CIS, 512 had MS, and essentially 839 of 901 had the Epstein-Barr nuclear antigen antibody. So that was 93.1%. Now, taking the rest of the people that didn't have that, 62% had, or 76 out of those patients who didn't have that, had the IG antibody to the VCA. Out of the remaining 17, all 17 had IgG antibodies to the EBV as detected by undiluted sera. So they kind of discovered in this trial that if you looked at Epstein-Barr sensitively enough, essentially you would run into, it would almost appear that every single person with MS was exposed to Epstein-Barr and had Epstein-Barr in their body. So there's also brain autopsy studies that demonstrated that Epstein-Barr was in a higher concentration in cells that were suggestive of Epstein-Barr infections were more prevalent in MS patients than the healthy controls. And also expression of Epstein-Barr seems to be in higher intensity and higher rates in people with active disease as compared to stable MS patients as compared to controls. They also looked at, this is another study where they looked at Epstein-Barr and essentially the various levels, and once again they detected a difference between the EBNA1, the VCA, and a difference in between RAS, CIS, and healthy controls. But they actually did not detect differences in the Epstein-Barr DNA and saliva. So it kind of suggests that maybe these cells that harbor the virus, that produce some of these proteins in the latent stages, actually predict this harbor population of Epstein-Barr cells that better predict ultimately demyelinating disease or other autoimmune diseases. So this is the big study that just recently came out. It includes 10 million US veterans. So the VA is good because every soldier that comes into the Army gets blood work. They do blood work throughout their service. And they were able to essentially, over a 20-year period, evaluate the risk of MS and Epstein-Barr. So out of the active duty US military, 955 patients developed MS. And it looks like the risk of MS was 32 times greater after being affected by Epstein-Barr. They actually compared it to cytomegalovirus, a virus that's spread similarly to the Epstein-Barr virus, in order to just make sure it wasn't just a random viral effect of maybe behaviors of people sharing foods or certain cultures. So essentially, the EBV is A and C. The CMV is B and D. And we'll see that very clearly that those who went on to develop MS, they seroconverted at a higher rate and had a greater risk of MS compared to those who didn't. And we saw no relationship with the CMV virus. Once again, something that we're starting to use in the clinic and will probably be used a lot more in clinical practice is something called neurofilament light chain. Neurofilament light chain is a protein that kind of is released by neurons when they're damaged. So MS has a lot of times when neurons are being damaged, so it gets released in the blood. And it looks like in individuals who essentially seroconvert, sometime after they seroconvert, they actually will have an increase in their neurofilament light chain compared to those that don't, which would also suggest that with one of the earliest studies that suggests after the infection, the risk of MS goes up, this would further kind of support that fact. So one of the reasons why we think that Epstein-Barr and MS might be related is there's a protein that is very similar to the gliocam protein that's on glial cells in the central nervous system. So we think that there might be molecular mimicry between those cells and essentially Epstein-Barr that tricks our immune system into damaging ourselves. So does Epstein-Barr virus, MS, and treatment? Essentially, there have been many trials that have looked at essentially Epstein-Barr and viral replication rates and MS patients. And essentially, for the most part, there's always a clear distinction between healthy controls and MS patients and some of these rates of these various viral markers. But looking at the interferon, glutaminyl acetate, and natalizumab or Tisabri, there wasn't a significant difference in these viral markers while on these medications. So a study of 30 ocrelizumab-treated patients, and ocrelizumab, as I said, affects, as you may or may not know, affects B cells, and I stated earlier that B cells are one of the cell types that gets infected by Epstein-Barr. They said that 24 months after essentially ocrelizumab treatment, several of the viral markers decreased in patients who were on ocrelizumab. So this could theoretically suggest that you're targeting those cells that harbor the Epstein virus that may trigger the disease of multiple sclerosis. So the question would be, could you actually prevent multiple sclerosis? And there's a theory that if you would vaccinate people against Epstein-Barr, and Epstein-Barr is the thing that actually causes MS, could you prevent the disease from occurring? And that's an area of active research that people are exploring. So there's there's a few different vaccines in development. So there's one that's currently being worked on that goes after essentially the protein that's responsible for its ability to attach to your B cells. It did show a reduction in mononucleosis, but it didn't prevent infections. Another concept is similar to how they're using vaccines to potentially treat cancers, is that they could potentially create a vaccine that would have your T cells better target these B cells that are harboring the Epstein virus and therefore clear those. So that's something that's been shown to be effective in cancer, as I said, and it's something that is being looked at to potentially use to treat multiple sclerosis to help better clear these EBV harboring B cells. Why I'm not 100% confident we'll ever be able to completely eradicate Epstein-Barr is about 15% or 50% of the population will have Epstein-Barr by the age of 5. So essentially you would have to vaccinate people from roughly 2 to 3 years old. And ideally, you'd probably have to do vaccination as part of the vaccination series that we do in infants. It's still even possible to transmit this virus vertically from a parent to a child. So that would be the mom gives the child the virus while in utero. With that said, it is something that if potentially could be curative or preventative and end MS in the world, but once again, we'd have to come up with a vaccine that works, prevents the infection, and then in turn, we'd have to inoculate enough of the population, similar to what was done with smallpox, to eradicate the virus from humanity. Once again, not only does it look like if we could prevent this virus, would it decrease MS or maybe even completely eliminate it, it could also eliminate several cancers, and there's also some associations with diseases like Parkinson's disease that this could also have an effect of decreasing the rate of it. So I know I have rambled for quite a bit of time, and I'm going to open up the floor to questions. So repeating the question, I was asked if I got like an Epstein-Barr titer, would I always pick a B cell depleter? And that's kind of one of the reasons why we think that Epstein or B cell depleters have strategically done well in areas that other MS disease modifying therapies have not. We could get into the weeds about why one of the B cell depleters worked in primary progressive MS where almost every other treatment has failed. And that's one of the theories, that there's these pockets of these B cells that line the surface of chronically inflamed lesions in the brain. They also line the surface of the brain in what we call extralymphatic tissue that kind of causes inflammation on the brain surface and damages the brain. That if you could target the B cells that live in these chronic lesions, maybe you could stop that, and maybe that's the reason why we see such an effect on progressive disease with therapies that target those cells specifically. So that is one of the theories. I mean, obviously it's not the only thing that does that, because if that was true, then we would see no disease progression with a CD20 agent, and we still do see that. But it is one of the theories that if you could target those cells specifically even better and affect them more, maybe you could stop the disease.
My understanding is the way that Ocrevus attacks or affects B cells, it kind of knocks things down and they come back over time. First question is, when they come back, would those have the effects from EBV? And over time, the longer you're on Ocrevus, does that increase or decrease the effects of the virus because you have potentially less and less B cells that are affected by EBV?
So theoretically, when the B cells come back, typically they're less inflammatory. So it's one of those things that I don't necessarily have the exact data in front of me. I know that I've seen it multiple times in the past. It would suggest, and when it comes back, the kind of protein markers that make up these B cells once again are less inflammatory. I think that we're kind of getting back into Epstein-Barr virus being the cause of essentially multiple sclerosis, or like the primary driver of it. I think that's going to be something that actively research is going to be done more. Now, one of the problems is that, okay, you get rid of the B cells, you target those B cells, but you don't necessarily kill every single set of the B cells. Like if you could get rid of all of them, then you would completely eliminate what was harboring essentially those B cells. So the problem is that you can get rid of probably about 99% of them, but that 1% that still exist, they can eventually kind of repopulate and essentially shed the virus again and affect other B cells. So that's the reason why, in theory, if it got rid of all the cells at once and you got rid of all the harboring like B cells that had the Epstein virus inside of it that were chronically infected, in theory, you wouldn't need ocrelizumab again because you got rid of all those cells. So it's one of those things that if you could to selectively target those B cells, because you can't get rid of all of your B cells because they are there, they do some things. But if you could get rid of like all the ones that were infected with the chronically infected Epstein-Barr, you could potentially eliminate the disease. Once again, chronically resetting it probably does a good job at kind of limiting those cells and their activity. So therefore, that's probably why we see the effects in progression. But once again, this is still musing that we're 100% correct that it is Epstein-Barr.
That's excellent. Thank you for walking us through a really complicated topic. And so I guess the final question is, do you think EBV is the sole cause of MS?
So I kind of have a feeling that I think it's a critical step. I'm not 100% convinced that it's the only thing that can cause it, because there are other things that can cause mononucleosis that isn't Epstein-Barr. But I'd say that the data kind of suggests, if you look hard enough, that there were some researchers even in England that would suggest that if you look hard enough and you don't find Epstein-Barr virus in an MS patient, you should start thinking of differential diagnosis. So I think that it's highly likely that it is something that is probably critical. I wouldn't necessarily go so far as to say necessary, but I'd say it looks like it plays a very central role in the disease.
Well, thank you so much for a wonderful presentation, and thank you to all the speakers who presented today, and for all of the folks who joined us in person. It was great to be back in person and see you again. And then for everybody online, thank you as well. We'll look forward to doing more of these, so stay tuned, and I hope you have a great rest of the weekend.